thank you for inviting me as well at first. I am Stefan Popp. As I just told you, I was at the University of Arizona doing my PhD, and that's what I'm going to be presenting on for the most part today, and search, and how it is not just Markovian random, and how it's not all coordinated through pheromones. Yeah, so I am going to be focusing on here, the biology. And so there's almost going to be no math in here. And I divided it up into 4 parts. The last 3 ones are chapters from my dissertation. The first one is more of a literature review of the search for targets of which you already know the destination. So imagine you are on this wide field here and you're in a tent in a home base, and all the way to the top left there is a mushroom that you are supposed to find, one of many that are out there on this field. So the question is now, when you have found this mushroom, how do you find your way back home if you cannot see the tent? How do you know in which direction to go? And also when you go out for the next time and you know roughly where you found the mushroom, but you want to find it exactly again out of this mushroom patch, rather, how do you find back out again? And so this is what a lot of the literature has focused on in ants. And this is very similar to here some ants that are living in northern Africa in the salt flats. So this is what they're seeing. There's basically no panorama or landmarks around unless there are some scientists putting down black cylinders there. And so the question is, after an ant has searched for food on this blue track here, and she has found some food, then she goes back to the nest relatively straight. So how does she do this? And the answer to that is through path integration. So basically they are integrating or adding up all the vectors of the steps that they have taken, so the distance and the turning angles between every step. And we know this because when you displace an ant from the feeder, so when you know that she is about to head back to the nest, you need to displace her by a few meters to the side, then the ant will go into the same direction, so in the cardinal direction that she expects the nest to be in, and then she starts a local search around there. She doesn't go to the actual nest direction. So it's idiothetic, or it depends on the information that the ant has in itself and not from anything outside, like landmarks. And we know that they count they count the steps because when you change the length of the legs by either cutting them short or putting them on stilts, then they, depending on how long their legs are, they undershoot when they're shorter, or they overshoot the target nest. Depending on the leg size. So they're not doing it through optic flow, for example, like other bees or other insects also do it. Although if you put these ants into a landscape with a lot of landmarks, and they also use other cues like optic flow to find the distance to the nest. Okay, so this is kind of classic ant navigation. Yes. 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 Yeah, good point. So basically all of what I'm going to tell you about here is individually, solitarily searching ants. So there are a lot of ant species out there that don't form pheromone trails to recruit. So it's all based on the individual. And so the foraging success is based on the search success of any individual ant. And then this homing is not perfect. So sometimes they also get blown away by the wind. And so when they are roughly at the position where they expect the nest, then they have to do a local search. And the way that they do this is by an increasing spiral around the point where they expect the nest. And this in different desert ant species, and on the left, even a pillbug or a woodlouse. And they are always like searching in spirals, but they interrupt these spirals by going back through the position where they expect the nest to be. And another one is here on the right, Demethorax illopenis. That is a an ant that is very closely related to the one that I did my studies on. And here, this ant is doing a search after an interrupted tandem run. That is because these ants, they don't form pheromone trails, but they do tandem running. And that is when, in this case, this ant is the leader, and she has found, in this case, the location of a new nest, and she physically leads just this single follower ant to a new nest, and she has lost the leader for a second there, searching around for the leader again, and the leader always waits to be touched by the follower. Yeah, so this is an instance of teaching, one could say, in animals. And yeah, so if you take away the leader ant, then the follower ant will start searching first randomly, and then, in a Brownian walk kind of fashion, and then expanding out with time. And yeah, this kind of search also depends on other factors like intrinsic factors or extrinsic ones. So here, for example, on the top, you see these ants doing the search for the nest, but then also when they have found a big food resource, which is not often the case, then they will also make repeated trips to that food source, like in this case, feeders. And it is different how they search around this location depending on what it is.
Sorry,
yes.
Can you just explain more about what
right. So, for example, here, this is the nest, and here's the location of a feeder that was there once, and then the feeder got removed, and the ants are going out again for the second time, and then they're searching around, and the heat maps are the redder, the more time the ants spent there. So you can see that here it's a little bit more centered, more focused when there is a lot of food, like the feeder was very plentiful, than in the middle case where there is not so much food and they're a little bit more dispersed. So they are adapting to that. The top is when they're coming back to the nest, and they expect the nest to be there, but they were displaced, so the nest is not there. And on the bottom, it's for food. So when they're going out to a feeder that once was there. Yeah, and if the ants are more experienced, so on the second or third time that they are either going back to the nest or going out to a feeder, they will also become more focused when it is in food, but not when they are more experienced in homing and going back to the nest. So that there is a different mechanism there as well, where maybe even on the first time that they're going back home, they already know really well how to search for it, but I don't know. Yeah, the same is the case if they're foraging for different types of food. So carbohydrates here, where it's relatively focused versus protein. And that makes sense because carbohydrates is something like aphids. So little insects, they produce honeydew. And so the ants are going back and forth again and again. This is very stable. While protein, you wouldn't expect that to stay in the same place because it's like dead insects. And if you have taken away the insect, then it's no longer there and it won't replenish. So they have evolved different kind of strategies for the different things that they are looking for. I will come back to that later as well. Yeah, so in summary of this first part, ants find back to the nest or food through food sources through path integration. They do spirals around the nest or known food resources, and they are doing more focused search with experience and more stable food sources. Okay, next part. Imagine now you are actually not searching for a place where you know where you want to go, but you're searching for some resource that is distributed in some way, but you don't know where the individual targets are. And therefore, I go over the general methods of my PhD thesis. I work with these ants, rock ants. Yeah, they nest in rock crevices, hence the name. They are relatively submissive and slowly moving, so they're not aggressive. And they forage for things like this. This is an aphid that the ant has just scavenged or maybe hunted. We don't really know yet their complete diet. and they don't have trail recruitment. As I said earlier as well, that means that the foraging success of the colony depends on the search success of the individuals. So that's something that they should have to be really good at. Yeah. This here are larvae. So the second stage, this is an egg. I don't know whether we find, no, there are no pupae there, but yeah.
But then so
This is a nest that was cut, like broken open.
Yeah.
Yes. Yes. There's no king there. Yeah, that's only in termites the case. So there's only the queen and the workers, and they're all female. And sometimes there are multiple queens. Yeah. Yeah, and these ants search for resources like water or new nest sites, which is really important for them. So this could be a nest site for them for a whole colony. They have very few individuals, or other invaders or attackers onto the nest that are from the same or different species. Yeah, or of course, food. Something interesting about them is also that they leave behind footprint pheromones. So they secrete some chemicals, some pheromones, and they leave them on the ground like footprints that are just smelly all the time. They can control it more or less, but we assume that it is basically like a footprint, that they always leave something behind, but they can up or down regulate how much. And yeah, probably passively all the time. Yeah, I'll skip that. It's not that important. Yeah, I get those ants, whole colonies from an area like this, a habitat with lots of trees. This is a whole colony that I'm just sucking in. Yes, I mean, there is a gaze there, so they don't go into my mouth usually. A lot of dirt usually, yes. Yeah, and then this is a whole colony in an artificial nest. Yeah, 50 to 300 ants per colony, and then I placed this artificial nest in a tracking arena. It's now somewhere underneath here, and so I record them then for 5 hours. All the ants can go out at the same time how they want for 5 hours, and I record them with cameras. And I track them. There is no individual identification of the ants between tracks because that's what they look like. So you can't really tell anything. You can tell one ant from apart. I don't think so. I've talked to Ian, and this is actually from Ian's. So the tracker that I'm using is from Ian's lab. Yeah, it's too few pixels. Yes. Yeah. Yeah, this is a huge arena here, undergrads for scale. So this is about 1,000 times the length of an ant. And there are no resources present, so I want to only look at the default search behavior. And then over these 5 hours, this is the kind of data that we get. A lot of ants, around 30 out at the same time, more or less. And over 5 hours, they cover a lot of the arena. Yeah, this is my artistic side. Yeah, and so the first question that I'm looking at is how, as I introduced already, how does the search behavior change with increasing experience? So after you have already learned the area and you know roughly where the mushroom patches are or not. And this is also really important for this species because they move their nest a lot. So they don't build a lot of their nests. So what you can see here in red outline, this is what they build, but otherwise they're just within the rock crevices. They also move when they are being disturbed, which can happen a lot if you just bump onto the rock. They move into a better nest site when there's one around. They move closer to resources when they are moving, and they even split their colony seasonally so that in the spring they move into different colonies and then they come back again in the fall. So that means that they move multiple times per year. And every time after they have moved, they need to familiarize themselves with the new surroundings. And so the question is, how do they do this? How does this change their movement? And for that, I filmed them not only over one day, but over 3 consecutive days. And here are visually the results. This is on the first day, then on the second day, and here on the third day. And what you can see in comparison between the day 1 and 3 is that on the first day, there are a lot more ants out, but they're clumped close to the nests in about a radius of 1 meter. And here on the third day, they're just more spread out, and there are fewer ants that are close to the nest. Yeah, and these movements that are of these additional ants that are there on the first day, close to the nest, they are also curvier and slower. And so this means that there's probably some additional familiarization going on, or maybe they're marking their territory when they're going out close to the nest. Yeah, and this is probably not driven by chemicals, but footprints, but probably only visual, because between the second and the third day, we exchanged the paper floor. So there is a new paper there, and we still see the same increase or increase of straightness or decrease of ants near the nest. So they probably need to familiarize themselves with the landmarks around them, around the nest. And this is how they, yeah, how they do this. Yeah. Yeah.
If they were still there when they, like hadn't changed the paper, they just ignored the ones that were left, like
pretty much, I think so. More or less ignore. Yes. We don't know it from this species, but from other species that have evaporated footprints, have a slightly different effect on them, but it's not really that strong because it's covered by all the ants. So there should be footprints everywhere that are old. Very blurry. They don't have a high visual acuity, but they can see like canopies or like in the room, like they can see the door and stuff like that. But I think nothing really small or even close up, it might be difficult for them. Yeah. I'll skip that conclusion. So with increasing familiarity, there is less activity far from the nest. The movements close to the nest are typically slower and curvier, and the change is not only due to footprints. And I have 3 hypotheses for that. That is, they are either patrolling for risk in the beginning, or they are memorizing their surroundings, or they are reacting to resource scarcity. So that means if they are out on the first and the second day and they see there is nothing there to be found, then maybe they don't go out, at least in the vicinity of the nest anymore.
Do they have any cache of resources?
They were starved for 2 weeks, but they're extraordinarily starvation resistant. So because they are so slow, they have a lot of like the fat metabolism is really slow, so they can store a lot of energy still in their bodies or in the larvae, for example. Yeah. All right. Then second part.
Can I just ask?
Yeah.
So did you try to compare the coverage time? Said you're going to find an area. So did you try to measure that? Time it takes for them to go around in different areas?
No.
You say that they can store in the book here, so
yeah.
Even a close environment and actual, I think that how long did it take to cover the time? So they would actually have. I don't know. I'll have to see what you've done in the paper exactly. Slow and curve, it might be very useful. I have to say, the first thing that comes to my mind.
Yeah. So there's basically a population or a subpopulation of these ants. So I don't know, 10 maybe, let's say, that are going just straight away on the first day and on the third day. So there's no difference there. And there's only more activity close to the nest of like additional ants going out. So I would assume that in total there is going to be a similar coverage of the area, but it's also difficult to say because I am not very interested in the behavior of the ants that are bouncing into the wall and then coming back, so I excluded them. And there are always weird wall effects if there are more ants out. Of course, they are going to cover the area more, but you know, how important is that for the ants or for the colony? I mean, they're going to cover the area more, but also they're going to oversample it. So
Oh yeah. No, but I'm saying that if you don't know anything about the environment, like a blind searcher, then if you are efficiently covering, then essentially you find out what information is out there.
Yeah.
So that becomes, because there are no clues whatsoever.
Yeah.
In terms of whatever they search.
Yeah. Yeah. Okay. How does the strategy change when you are in a search team and you have to coordinate with these other people that are searching with you? Maybe you're thinking you can look at the footprints that the others are leaving behind in the grass, and yeah, that is also what the ants might be doing. That means they can also sense where others have been by the footprints that the others have been leaving behind, and how much there has been accumulating. And so they may be able to use this to avoid where others have already been. And previous work shows that, yes, if they have access basically to the footprints of the other ants, then they will search more evenly than if they are just searching individually and you to basically add all the individual ants up. So they must coordinate somehow. And I have two hypotheses for this. One is that the ants turn away actively from the footprints. So when they smell more footprints on the left antenna, then they turn to the right side where there's less. Or another hypothesis is that the ants are moving straighter when they are on areas with higher footprint concentration, so that they're basically dispersing or diffusing away quicker from these high footprint areas, and then they're just walking more curvy when there are less footprints out there. I do not find evidence for the first hypothesis that they're turning away from the footprints. You can see that by the very straight line here. So they are not turning away, which is on the y-axis, turning into one or another direction, depending on the difference between the concentration of footprints on the left and right antenna. As a side, that is a proxy by calculating how often an ant has walked over that point, basically pixel, before. So it's just a number of ants having walked before. It's not going to be exactly the chemical concentration, but it's a proxy. It's the best we can do because they're just tiny amounts, and we cannot really assess that better. Yeah, and this also kind of makes sense because other people have shown with robots, these kilobots, that avoiding the tracks of others, also yourself, is not that efficient when you are searching with a lot of other individuals. Yeah, well, how about the second hypothesis, that the ants are diffusing away quicker? We also don't find evidence for that. As you can see by the negative slope here, so the more footprints there are going to the right, the less straight they become. So this is actually the inverse of what the hypothesis states. So this brings up the question why. And there are two reasons for that I have identified. One is that there's a combination between two factors. There are fewer footprints that are farther away from the nest, which is obvious given geometry. So the redder, the more footprints there are. And secondly, there are some ants that move straighter and they move farther away from the nest just intrinsically. And that leads to a situation like this, where these ants that are curvy to begin with, they stay closer to the nest. in the same time or distance walked, and then they automatically are also staying in higher footprint concentrations. While those that are walking straight, they are more likely to go farther away and thus into areas of fewer footprints. So it's a kind of an inverse relationship. This is just showing again that there are some ants that are walking straighter. They are actually also going farther, which you would expect from a random walk. Yeah, so in conclusion, the reason that ants are walking curvier on higher footprints is that straighter ants move farther away where there are fewer footprints. So the causation is that straightness influences on how many footprints you're walking, and not the footprints influence how straight you're walking as an ant. Yeah, and if we are controlling for the distance, then this correlation should vanish. And therefore, we bind the data into different distances from the nest. And then we have in this graph on the x-axis the distance to the nest, on the y-axis the slope of the straightness predicted by the footprint concentration. That means down here, a negative slope is what we find, or what we found in the beginning, that the ants are walking curvier when there are a higher footprint concentration. And on the top, that is the inverse, they are dispersing away quicker on higher footprint concentrations. What we see are negative slopes, which means that there's still this initial correlation that we found. So the distance to the nest does not account for that negative slope, so of the ants doing the opposite thing that we would expect them to do. How is that, or why is that? It's because some areas make ants walk less straight and thus deposit more pheromones there. And what I mean by that is that you see here in this black oval, there is a horizontal line of green, which is generally lower straightness. And also on top, there is, you can see it here, there are horizontal red lines with more footprint concentrations. And those areas or lines coincide with where there was adhesive tape, like 2 or 3 layers of paper beneath the layer that the ants are walking on. And so I wanted to make sure, that they're not smelling anything off that tape, but still they could probably smell that tape. And that is responsible for these, the patterns here. And since this is under like 2 other papers of layers of paper, I think it is the smell. That's the only thing that I could think of that they are sensing. I mean, it might be the tape is plastic, so there might be electro things going on there that they are sensing. But yeah, it is something. Even though, of course, they are very, very sensitive to the height differences as well. I also think that it is the smell, because I'll use a pointer. Here in the middle of the room, you see, I don't know whether it's human seeing patterns or not, but I think this is a circle where there is more blue in the middle. So where the ants are walking straighter. And this is also the place where there was a drain in the middle of the room, and there were some smells coming up from the sewage, basically. So something I had never covered. It was covered underneath like cardboard and PVC and paper, but still, there is this pattern there. Yeah. All right. Oh. So that means that oh, I missed that. Anyway, so this means that they are paying a lot more attention to the smells and what's on the ground. And of course, if they're in the forest, on the forest floor, then there will be a lot of rubble and gravel and stuff like that. So it's going to be a lot more important for them than reacting to the footprint pheromones of their nest mates. And so that means that they are coordinating with communication, probably, so that because others have found that they are dispersing more when they have access to the footprints, that they are probably forming an area where they want to go, so to speak, by reacting to where other ants have been and are, and they are keeping that as basically as sectors instead of reacting ad hoc to the footprints that they are currently encountering. And so this is something similar we see in other species that are also, other ants, that are also hunting or foraging solitarily. And you can see a really clear sectoring. Those are only like 5 or 6 ants that are foraging, and every ant has its own sector, basically, more or less. And that's also something I saw in another side project that I did with these ants. They are really cool because they do wound care. So when they are going on raids and they're being injured by termites that they're foraging on, then they have here, for example, this is a leg stump, and then another ant, this is a paramedic ant, basically. It licks the wounds, and then it has a higher survival chance in the next raid that they're going on. Anyway, so I was trying to film those or follow the scouts that are scouting out where to do the raids of these termites that they are foraging on, and I yet have to be able to track those ants because it's really difficult with a moving camera and with all the grass. Conclusion to the second part or third. So there is more even area coverage through inter-ant variation and not through direct communication. There is more even area coverage through the large scale avoidance of footprints, probably. And there is an extreme sensitivity to the environment, and this is probably more important than any chemical footprints.
So the direct communication would be what? What is the potential direct?
Direct is even just smelling the footprints of another ant that has just walked in front of you.
Well, that you call direct communication.
Yeah.
Oh.
Instead of
I would call it indirect.
Well, yes, direct or indirect communication, I guess. Yeah. In the extreme case, they are not communicating at all. That's kind of the current hypothesis, that they are not communicating, and they are forming their own sectors or areas by basically choosing the area where they have already found the most food in previously. So they become basically superstitious, and they always go into the same area where they have found the most food. And if the food is or somehow distributed, then it means that some ants are going to favor one area over another, and that leads to a sectoring out.
So you're saying that, often we hear about, ants, they coordinate, stick merging.
Yeah.
Then you're saying they're just independent. It just happens that was just say.
Yes.
Wow, okay.
And that is because there's a wide spectrum of ants. So those that we hear about the most, those are the ones with big colonies. And there it makes sense that the stigmergy, and like Argentine ants, for example, they have basically no individual behavior, and it's all just what happens in between the ants. But here, like with those examples that I've shown, those are 300, maybe 1,000 individuals, and there the individual has a lot more individuality, I guess. And so it depends less on the communication and coordination, like directly with other nest mates, and more that they are just more individuals that are working towards the same goal. And yeah.
So what do you think is more efficient?
Well, it depends on how big the colony is. That's
so you're saying that once the colony gets bigger beyond a certain size, then you bet really coordinate.
Yeah, there is this hypothesis that has been there for 30 years or so, where we observe that the smaller the colony is, the less communication and coordination there is. And so we do see that trend with increasing colony size, even like between species and also even within species. That in the beginning of the founding 3-4 foragers, they have a very different behavior than the ones that are coming when there are already 1,000 or 10,000 ants out there. Yes, like there are different foraging systems, like the one that's the mass for recruiting, where it's the typical trails that you see. But then there's also group foraging, where one leader ant is taking out maybe 10 to 30 ants on a short-lived foraging trail to a place that the leader has just discovered. And then there's the tandem running that I just showed, or then no recruitment at all. And so you find them in different species of different typical colony sizes. Yeah. All right, to the last and maybe most interesting part. Again, what is your strategy? So what is your actual movement pattern that you will adopt when you are alone searching for mushroom patches? Maybe you think you should search in a systematic way, like in a spiral or in a zigzag pattern here. And yeah, the question is, so these are the two possibilities, in either a systematic or in a random way, in a random walk. So the question is, how systematic should we search? What is most efficient? And in a mathematically ideal world, the systematic searches are best because they cover each spot only once, but they cover all the spots that are closest to the nest. And in the random walk, you have these two criteria not given. But in the real world, you have to contend with obstacles in the way and with navigational uncertainty or wind blowing you away. And then these systematic strategies might become much worse, and even worse than a random walk. And there is some simulation evidence for that. So if there's no noise, then of course the spiral is ideal. But then if there's some noise or even more noise, it becomes really bad and even worse than a random walk. Because in the random walk, this kind of noise is already baked in and it cancels out, so to speak. Yeah, so then there is another question that is, what do the animals do? And in the literature of, in biology literature of walking animals or searching animals, it's always or usually assumed that they're doing a random walk, some sort of correlated random walk, and that's how they are analyzed. But I want to know what these ants that I'm researching are doing. And they are actually doing meanders. So not a pure random walk, but a systematic strategy where they are always going left to right in some kind of curves. And so on the black, that is a real ant track, and on the bottom, that is a simulated ant track that is made from the same characteristics, walking characteristics of that real ant. To give you a sense of scale, here is the whole arena again, and that is the example track. And if you zoom into this point here, you will see an ant here in the red circle. All right, so how do we test for this non-random meandering? And I mean, we see that it's probably meandering, but I also want to give evidence for that. So first of all, I created this random walk null model, and then I measured the turn autocorrelation. The random walk null model is just basically shuffling all the individual frames or like steps of the ant track around. And so that gives us the same distribution of turn angles, but the steps are just scrambled, so without any potential meandering. And the turn angle autocorrelation is 1 if the ant is going into the same direction or straight all the time, or between two points that we are measuring, and it's -1 if there's perfect zigzagging. So if the autocorrelation is below 0, then there is some kind of meandering going on.
Sorry, the zigzagging means what?
The negative autocorrelation means zigzagging, and that means it would be the like exactly -1. That would be the path. Yes.
So it's just between the left path, but still going somewhere.
Yes. Yeah.
So it's not persistent. So that's, you use the word persistent with it, which is related to the turning angle, no? So big turning angle, you're persistent. It's completely flat, it's random, no?
Yeah.
And then if you actually make it anti-persistent, then you keep back and forth. So I'm trying to understand the nomenclature you're using here.
Yeah, that is so the distribution is it's so the autocorrelation is between 2 points that are following on the same track. Right, so you're picking one starting point, it goes to the left, you just pick another point like 10 steps down the line, and you see whether this point goes also to the left or to the right. And if it always goes to the left, like you go through the track and this sliding window, and if this point goes to the left and then 10 points later, it always goes to the left, and if this one goes to the right, this one also goes to the right, that's autocorrelation, then you have a perfect autocorrelation of 1.
So you're coarse graining the temporal, so because they usually take turning angles, no? Because sort of coarse graining that counting just left to right, no? And then okay.
And related to that, what you are measuring is you have some resolution in the tracking, which is it the same as individual? I mean, are you watching individual steps or some coarse grading of that?
Pretty much individual steps. Yeah. So it's like 2 millimeters. Yeah.
So it's funny, if I were to ask you, take a track of one individual and then you plot the turning angle distribution.
Yeah.
Right. What do you see?
You see
away from the walls, let's
say. Yeah. You see the typical whatever it is, peaked around 0. Yes. What this does, autocorrelation, is it splits it basically up. You take one point that is somewhere on this distribution, and then you see, okay, 10 points down the line, what is the distribution of these angles? So it's going to be a subset of all points, right? And then this one is going to have a slightly skewed distribution. Like if you take a random step from this distribution, a random angle, and then you ask, what is the angle of the following step? It's going to be, because it's a correlated random walk, it's going to be somewhere peaked around where you, I don't know.
Yeah, but then the reference point is, that's why they use the turning angle. You always relate to the previous step. So what's your direction you came from?
Yeah.
And then it becomes natural. Okay, I'll ask you later. I just wanted to understand. So it seems some sort of, on one hand, you sort of coarse grain because you just talk about left and right. On the other hand, you try to talk about in time what's happening. That's the part I'm not really understanding, but that's okay. So
okay.
I can ask
yeah. Jesus. Okay. What we see with this measure is that 80% of the ants do meander significantly, and so there are also some statistics associated with the autocorrelation measure. Yeah, but there are also some ants who don't meander, so it's very widespread. Yeah.
So the meander when you became.
Yes. Yeah. And it's really interesting that this is found not only in these ants, but across the tree of life, basically, from other ants, but also moth larvae, limpets, bark beetle, but also only some. And Dictyostelium, a slime mold, which is also congregating together, but doesn't even have a brain or any neurons. They also do a zigzag. Yeah, and in the bottom left, this is paleomeandron, which is something similar to that. A worm about 450 million years ago that was slithering through the mud of the ancient seas. And so this is also fossilized, one of the oldest behaviors that we know of. And what people have been doing there is pretty much just describing them with these more or less subjective ways of characterizing them. And interestingly, also the same has been done for rivers that are also meandering. That's where I got the name from. But again, there is not a lot of quantitative or even mechanistic understanding of how. Really? Huh. Yeah, so there are also meanders found in other places. And I would be really interested to see like a unified theory of meandering, like what makes it and yeah, why? What are the differences between different yeah, scenarios? What I'm more proximally interested in is a mechanism of how any ants are doing this. And so there has been some work 30 years ago by people who tried to find out like how these ancient fossils were doing this, and they were describing a mechanism with just 3 or 4 different behaviors. Very simple. One is phobotaxis, that is avoiding the trace that you have already made. The second one is thigmotaxis, so following your own trail that you've already made. The third one is strophotaxis. So this is just more or less randomly turning into one or another direction away from where you've been. Yes.
That use, okay, but following the boundaries.
Yeah.
So why do you?
It's not only boundary, it's any longitudinal thing.
Oh, I see.
Yeah, and then they said, okay, you can really easily make a model of someone that turns like this by having just 2 pairs of sensors, and then given like which sensor is on the trail, you can change the behavior of the robot. So they made this robot, and the trails were toilet paper that this robot was dragging behind itself, and it was qualitatively speaking recreating these tracks of the worms relatively well. They, yeah, qualitatively. Those 3. So when the robot is close to its own trace, then it follows it until some predetermined like stochastic time when it alternates and when it turns around, and it turns until it finds its own trail again. Yeah. And if you can change that time or that clock, and you can produce different kinds of patterns from spirals to these meanders. Yes, for rivers, no. That's true. Yeah. So it's not as simple, as I will also show. Yeah, but it can do something that looks very close to what these worms were doing. Yeah, so it's not like my ants, because these ants, they don't even touch their own the trail that they've made that often, at least. Yeah. But in other desert ants, now those from Australia, they also show a very significant oscillation or meandering going left to right. So this is basically the same. This ant is going from the right to the left. And they are interestingly doing this in order to orient themselves where they are. So to look to the left, look to the right, and like compare their snapshots that they have made with they're currently seeing, to know whether they're looking into the direction of the nest or somewhere away, or where they are in general. And It's somewhat similar. So what they're doing is comparing. So in the beginning, when they're leaving the nest, and then they have to learn where they are and where the nest is, they turn back to the nest, and we think they take snapshots in memory. And then later, when they're going out and coming back again, they compare how well the panorama matches with what they're currently seeing. And they can turn left or right, and they see, okay, if I turn to the left, then this panorama matches better. So they will kind of go into that direction more. So in that case, it's kind of similar to visual taxes. Yeah, but it's a little bit more complicated. Yeah, and they also do this kind of behavior when they are on a tracking ball. So they just walk, and this styrofoam ball just rotates underneath them, and then you can also track them where they would have been going. And that means that they also do this kind of oscillation without changing the scenery. So that means that it's an intrinsically generated behavior as oscillation.
So, Stefan, so by just looking the way you've drawn it, so it seems like a very persistent, highly persistent, not really ballistic.
Exactly.
Overall line.
Right. Yes.
And then you perturb with left and right turns.
Yes.
So it's two modes, and then randomly, I mean, it's not that randomly, but then alternatively you go a little bit on the left, and then turn, and so it's two modes on top of each other. So one is nearly ballistic, and
yes,
the other one
yes,
you have to build in.
Yeah, right. In this case, it is almost ballistic because the ant is trying to get back to the nest. So this is I think that is a really interesting point, that they are doing this basically only, or also going back to the nest, but my ants are searching for something probably, and I don't think that they have to navigate by going left and right in order to find back to the nest, because they are also just walking outside and trying to find these like new novel food sources. So my hypothesis is that they are adapting the same mechanism of oscillation for two different purposes. And I will also, I have to skip anyway. Oops, here. This is a beautiful example of one of the tracks in my data set, where you see that they are turning left and right somewhat regularly, but in all kinds of different directions. And here, this is also another great example where, just like you say, on different scales, or like I think here on 3 different scales, they're doing the same left and right meandering. And so, yeah, this is really interesting. And I don't know how I would model how they're doing this on these 3 different scales, so that they are not overlapping themselves, like crossing their own paths again and again.
Yeah, there are a couple of cases where they cross there, but just 2 or 3 cases.
They don't cross this. It's just because of how it's painted. Basically, they come very close, but then they don't cross it. And this is the other. So this is the first thing. How do they do these meanders? But also, there's a lot of stochasticity in there still. It's not like with the desert ants. And another part is self-avoidance, where they come really, really close so that they can already smell their path, but then they turn the other way. I try to just give evidence for that, but it's really difficult actually to do it without an experiment, to show that they are avoiding their own footprint path. Yeah, I would be really interested in pursuing that, because if you have, so here in another simulation from someone else, if you have robots without or agents without evaporation, that are always turning away from their own footprints or their own pheromones, then they will get stuck in circles or in loops. And if you have evaporation of these pheromones, then you get a better coverage than without pheromones or without evaporation. So there must be something like that going on in the ants that they are forgetting or they are disregarding. parts of the pheromones that they have already laid. So this is, similar to self-avoiding walk algorithms, where they also have to avoid their own trails, but they do this in a, in some kind of way by checking. I mean, there are different algorithms, and I'm not an expert, but either through backtracking, which the ants can do, or checking on a global way whether they would close the loop or not. And I'm not sure how much the ants are doing this, but I think there is some algorithm that keeps these ants from crossing again and again over their own paths, while also having that small scale meandering mechanism in there, and while also having a bias to first go out away from the nest, because they have to go away, but then also not be too straight. Yeah, and so all of these different mechanisms are layered on top of one another. We are at the hour. Yeah, this is another ant species. This is the Argentine ants. They are very bad for wherever they are invasive. And yeah, this is an unpublished database that I just got access to from Simon Grenier. And I also would like to do something with that, because it's really striking almost how these ants are going in arcs as well, and then they are going back, like they go here in a 180 arc, and then going back again, and then on another arc, almost like garlands. But on the right here, this is another ant that just goes into circles, and then only sometimes reverses. So there's the same kind of mechanism where the ants are going, turning in one direction until some threshold, and then they're going back, or they're even reversing.
They are different ants.
Different ants, yes.
Same conditions.
Yes. So what I have in mind is that all these, or it is more, or it's somehow better to describe these kinds of paths in terms of arcs, like arc length and turning angle, instead of steps and turns. Because this is probably more the mechanism that the ant is setting out to do some arc in some kind of radius, or a meander, some kind of radius, and then at some point it decides to change that radius instead of going, step and turn wise. Should I stop there? I mean, I rushed through the last part, but
Yeah. Any questions? Good question. Yeah. Lots of phenomena.
Do you imagine that they have, we're thinking about the ducks filling the rectangular arena? Imagine that they have this, like this is something. They have, when they explode, they have these place cells, tiny brains, like mammals have. Don't have a program.
no. Yes, but they do have mushroom bodies, which is a completely different brain part, but it does analogous things. So yeah, I think I'm not sure, but I think there are indications that something like place cells might be happening in insects as well.
Mushroom bodies.
I think that's where it is. Yeah, so there are the mushroom bodies that are the integration centers, but it might also be the central complex, which is similar to the hippocampus, I believe. Yeah, there are a lot of analogies, just in different brain centers. Yeah. No, not yet. Yes.
Any other question? Okay, so let's thank you.